By the way, while we're here, let's give a round of applause for the audiovisual and tech people. I think they've done a really good job today. It's one of those unfortunate jobs. If you do it well, no one knows you're there. That's someone who's done it really, really well, right? So it's kind of it's like refereeing in sports. If you do a really good job, you're not noticed. So I want to start off by basically saying that this is a this is a teaching lecture, not a research seminar, because looking over the list of topics, I thought there'd be some useful tools to try to give to people. For starters, I would like to thank the organizers, Kavita, Kristen, and others. So what I want to do today, and so I also want to stress how this lecture is going to be structured. It's a bunch of little short topics that have a common theme. What I'll do is I'll, present some theory, some motivation, then take questions, finish that, move on to the next one. Also, some of this stuff initially seems a little bit abstract. I have some examples which I think will help you on things. And so I'm going to cover, for example, things to do with large sets of p-values, and also a little bit at the end on model selection. So most of you probably know about Bonferroni. And most of you probably know that Bonferroni is good and bad. It's bad because it's too harsh. So now we have this thing called FDR. People have heard about false discovery rate? How many people can explain to me what false discovery rate is and feel comfortable about it? Even Nick has his hand down. Don't feel bad. I'll hopefully be able to walk that through you at the end of today's lecture. So I want to stress that in the lecture, all the stuff I'm going to be covering, give me half a second. Here we go. Okay. So all the stuff I'm going to, questions or comments?
It was about the false discovery rate.
Okay.
So to me, it's the number of false positive divided by the total number of positive tests. And to control for it, there is some algorithm based on the rank of p-values.
Okay. And so when would we use that? When does it make sense to use the false discovery rate? We'll talk about this, but you might think about that. And the short answer is when you expect a small but non-trivial fraction of your test to actually be from the alternative hypothesis. So the bulk of what I'm talking about is discussed in Appendix 6, which is called Multiple Comparisons and Meta-Analysis. I gave Kavita a copy of that. It should be posted on Slack. I'll be talking a little bit about stuff from 21. So we've typically talked about a GWAS where we test each SNP separately. Well, there are other ways to go, and one way to go is called the gene-based GWAS, where I take all the SNPs in one gene and ask if their overall signal is significant. Or I take several genes in a pathway and ask if their collection of p-values suggests there's something there. So a lot of the stuff I'm going to be talking about, that's a good application of. I also posted chapter 27 from Lynch and Walsh. That's on adaptive walks, so you might find that interesting. But it also, on these 2 pages, talks about something called extreme value theory, which I'll use as a way to supercharge your permutations. So what's the motivation? Well, you've clearly seen by every single talk here, they're testing typically hundreds of thousands of associations or observations or tests. It could be a SNP, it could be scoring some molecular phenotype. With modern genomic data and functional genomic data, the parameter space is massive. So this raises serious concerns about multiple comparisons. If all the tests are truly from the null hypothesis of no effect, then by chance I'm going to have some of them register as being significant unless my criteria for calling significance is really stringent. So how do we handle that? How do we combine p-values over different sets of tests, such as the SNP in a gene or a series of SNPs on a pathway? How do we combine estimate of effect size over multiple studies? So people, when they are combining data, a meta-analysis means an analysis of analyses. When people are doing a meta-analysis, the hypothesis is almost never that this effect is non-zero. We're almost guaranteed that. The hypothesis is what is the best estimate of this effect over all my studies, and is there any evidence that the effect varies from study to study? So meta-analysis focuses on effect sizes rather than p-values because it's the more natural way to frame things. And in chapter 6, appendix 6, I give some examples and talk about why this is a better approach. Then I'll really briefly talk about model selection with high-dimensional data. So we'll start out with something a lot of you probably know, which is Hotelling's t-test. Then we'll move on to things like AIC, BIC, assessing model fit, and we'll briefly mention penalized regressions like lasso and things of that nature. So here's an overview. So the first part, I'm going to talk about control of significant tests over multiple comparisons. The most obvious question to ask is there an excess of false positives? Are there too many significant tests? If so, how would I test that? And this turns out to have a very interesting result. It allows you to estimate the number of hypotheses that are likely from the null. So if I'm testing 10,000 tests, did my mic just go out? No. If I'm testing 10,000 tests, it would be nice to know the excess number of false positives actually gives us a way to estimate what fraction of the tests are actually from the null. It doesn't tell you which one are, but it gives you a way to estimate that. We'll talk about that. Everybody's heard about Bonferroni, but there are methods like sequential Bonferroni. For example, SIMS is really good. We'll talk about the effective number of tests. SNPs are correlated. How do you adjust for that? Short answer, eigenstructure of the correlation matrix. We'll talk about the false discovery rate. We'll talk about Bayesian approaches. A motivation for Bayesian approaches is you've just done a GWAS and you've tested 500,000 SNPs. So you assign a Bonferroni value for that. You've done the test, they're done. All of a sudden now, a new imputation comes up. Imputation basically is a way of using a reference population and your recorded SNPs to infer or guess or estimate SNPs that are in between them. So now all of a sudden you now have another million SNPs that you didn't test originally. So your 500,000 tests that you did test, some are significant. Now all of a sudden, by simply adding a million SNPs, even without testing them, under the classic testing framework, some of those 500,000 SNPs now become non-significant. Why? That Bonferroni number on the bottom gets really big. That kind of doesn't make any sense. So Bayesian methods are a way to handle that, and they're actually quite nice. And then, because Kristen wanted, we'll talk about permutation tests quickly, but hopefully enough detail, and something called the extreme value theorem. And the motivation here is, typically a permutation test means you shuffle your data and you rerun your analysis. Well, that takes time. So if you're lucky, maybe you do 10,000. Well, that'll give you a p-value about 1 in 1,000 for your cutoff value. Suppose you're combining several different tests. So to adjust for multiple comparisons, that critical value is not 1 in 1,000. Maybe it's 10 to the minus 7. How do you deal with that with permutations? And the answer is extreme value theory. So we'll talk about that. Then we'll talk about ways for combining p-values from different sources. I have a series of tests. How do I combine those p-values? Fisher and Schaefer are the classic. Then we have Tippett, Sims, and the new upcoming really interesting one is the Cauchy. If you've had a probability class, you've probably heard about the Cauchy distribution as something that's really bad because its area is 1. It's a proper probability distribution. But all the moments are infinite. But it turns out it's actually very useful for some things in combining. This is a pretty recent thing. We'll talk about why that's the case. We'll talk about meta-analysis. Then we'll talk about fitting high-dimensional data. Again, fairly quickly. Penalized regression, model selection, AIC, BSC. So those are kind of the 4 chunks we're going to go through. That's kind of your roadmap to take us on there. So it's always good to start out with a central motivating idea, a way to think about potentially complex problems. So going forward, when you come to a problem like this, you'll have some insight, and that has to do with how we look at p-values. Before we do that, just some terminology, so we're all on the same page. A significance is a probability of seeing a test statistic as extreme as you see, given it comes from the null. So for example, a 5% significance would mean that I assume it comes from a null, let's say a mean 0 and a known variance. Then I go over to 1.65, I think, and if it's a unit normal. And if I'm above that, I call the test significant. So changing that significance level gives you control over how many false positives. That is, a test that has no effect that I still declare as being significant. We call that a probability of a false positive. The test level significant, or the error rate, also sometimes called the comparison-wide error rate, CWER, when talking about tests, a P of 0.05 means that if I take a random test that's from the null, only 5% of the time I'm going to declare that to be significant. So that's something we want to control. But if we control it too much, things which are not from the null don't have enough power to be detected. So there's always a trade-off. If I have a series of tests, the experiment-wide significance, also called the family-wide error rate, so now I use gamma. A gamma of 5% means over my entire series of tests, the probability in any one of those tests of seeing a test significant this extreme or more is only 5%. So I can test at an individual level, and I can put everything together and make try to get something over the entire set. And discoveries are tests that we declare to be significant. We have some significant criteria, and the test statistic exceeds that. That is, its p-value is smaller, and we call that a discovery. Discoveries can be good. That's true discovery. It's from the alternative, and I'll make the alternative simple. It has an effect not zero versus having an effect of zero, or it could be a false discovery. That is, there's really no effect, but by sampling noise, I declare it to be an effect. So just some terminology. Significance, discoveries. Now here's the problem. Suppose I test 100 hypotheses, each at 0.05. Well, how many things do I expect to be significant? Well, that's 5. I expect 5 false positives under the null. So in order to adjust that, I have to, instead of testing at p of 0.05 for an individual test, if I want a p05 over all 100 tests collectively, I've got to set some threshold level which is smaller, often significantly smaller. So I test each test with more stringency. That lowers my power. So there's always this trade-off between worry about making a mistake and worry about throwing stuff out. And that'll come when we talk about the false discovery rate and why you use Bonferroni in some settings and false discovery in others. Here's the key idea for a lot of them we're talking about. It's a very simple idea, and once you see it, it's obvious, but like a lot of great ideas in science, until someone points it out, you don't see it. Once they point it out, well, of course, everybody knew that, but you didn't 10 seconds ago. So if I take all my p-values, I've done 10,000 tests. Each test has a p-value. I can then make a histogram. If all those tests are from the null, the distribution is a uniform over 0, 1. So it should be a flat histogram with a little bit of fluctuation. The other way to look at that is I can rank them. The smallest p-value, next smallest, et cetera, and get what's called a QQ plot. And on that, as I go up higher rank, the p-values should go higher, so it should get a linear trend on a QQ plot or a flat trend on a histogram. So that idea, that critical idea, if your hypothesis is from the null, the distribution of p-value should be from a uniform distribution over 0, 1. That's used in a lot of ways very powerfully. Question? Yeah, microphone? Get microphone over here. Just was on the recording. Let me go back.
I don't understand the line that if all tests are from the null.
So suppose I'm looking at 10,000 SNPs, and suppose none of those SNPs have an effect. From the null hypothesis is that this SNP either has an effect or it does not have an effect. If it's from the null, I'm assuming it doesn't have an effect. If it has an effect, it's from the alternative. So we talk about the null. The easiest way to think about that is if the thing I'm testing has no true effect, and I compute a p-value for that and do that 10,000 times, that distribution of p-values is going to be uniform. It'll be flat over 0, 1. And the next slide will show some deviation difference from that.
Why would it be uniform?
That's what happens. If I have no effect, the p-value is going to be uniformly distributed from 0, 1, right? So maybe this will clear it up here for you. So here are simulated distributions, and I have 5,000 tests, and I have 25 draws from a normal with a mean of mu and a variance of 1, and my null hypothesis is that mu is less than or equal to 0. So this is a one-sided hypothesis. I did this for a specific reason. So here, if in block A, I assume mu equals 0 and then drew 25 draws from that, tested whether or not this is correct and computed a p-value. And this is that histogram of those 5,000 p-values from doing that test over and over and over again. And as you see, it's flat, it's a uniform. Now let's go over to panel B. Panel B, I assume that mu equals 0.2. I'm testing that mu is less than 0. And what you see is instead of this being flat, I have a huge excess of values near 0. Right away, that tells you don't expect that many. It should be flat. We'll talk about how you test for that. The reason I did a one-sided test, you can also see this over here, where I have an excess near 1. That sounds a little bit odd at first, but it makes perfect sense here. I'm testing whether or not mu is less than 0. I'm assuming here mu is minus 2. So when I'm testing less than 0, I'm looking at that tail of the distribution that's above 0. As that mean gets less and less and less, that 1 minus that tail, that is the area under here, is my p-value. So as I scoot off to one side, that p-value approaches 1. Because almost every single test is going to say, of course, you're less than 0 because you're negative 2. So we very often see a spike near 0, but if you do one-sided tests or other configurations, you can often see a spike near 1 as well. And of course, you could see a U-shaped distribution as well. The bottom line is, if you plot out all the p-values in your sample, and you often see this when people do a QQ plot, where it should be a straight line, there's a bunch of stuff, and all of a sudden, as it gets near 0, they really spike out. Well, those are likely significance. The rest is kind of just things that have no effect. So these distributions of p-values over all your tests have information in them. That's kind of the introduction. Before we talk about number of false positives over a family of tests, questions so far? Kind of terminology. The key issue is this idea that p-values, if I do a whole bunch of tests and each test is done under the assumption of the null, that is, there's no effect of the thing I'm testing, then the resulting p-value is going to have a uniform distribution. Or if I plot them by rank from largest to smallest, I'll get a straight line. That plot's called the QQ plot. So let's look at some application of this idea. How many false positives do I expect? So I have n tests. Where's n here? I have n tests. We'll call this trials because we're talking about your friend, the binomial distribution. By the way, in this lecture, you'll get about 7 different distributions, many of which you've seen, some of which you haven't. So let's imagine I'm testing each with probability alpha. So this is the probability of getting j false positives out of n tests when all are from the null, and therefore alpha is the chance that I declare that test to be significant. 1 minus alpha is the chance I declare that not to be significant. We'll show an application of that in a second. I can also, instead of using a binomial, I can also simply use a Poisson, where now my success parameter is the expected number of false positives. So what's an application of this? Again, you can see here, when I have like an A 6.5, this is just cut and paste from that appendix 6. So you have all that in front of you. So suppose I have 250 independent tests, and I test each test with a 2.5%. So what I'm saying is my cutoff value is I'll figure out the distribution of my test statistic under the null, let's say a normal with a certain variance. And then I'll go out and I'll find that value over which I only exceed that value 2.5% of the time, and I'll call that my significance. So in this, I expect 250 times 0.025, or a little bit over 6 tests that are false positives if all these tests are from the null. I see 15. Well, that's bigger. Is it significantly bigger? Well, easily just sum up by a binomial. So this is a chance of getting 15 or more tests given 250 tests, and given that's the alpha value, and there's the R command for it. It's easy to do. You do that, and you see that the chance of getting 15 or more tests when you expect 6.5 is about 0.2%. So it's clearly an excess number of false positives, which means that some fraction of those are not from the null, that is, they do have some effect. If I do the same thing using the Poisson and plug in 6.25, I get, instead of 0.18%, I get 0.21%. So you see how you can do this, really simple calculations to do, very straightforward. The interesting question is, given an excess number of false positives, what more can I say? And the cool thing you can say is you can estimate n sub naught the number of nulls. So what that I mean is I've got 250 tests. Each test is an independent test. How many of those are actually from the null? Is it 225? Is it 200? Is it 150? You can actually estimate that from the distribution of p-values. And people often want to look at the fraction of all, so n is all the tests and n naughts. This is the fraction of all the tests that are from the null. So spoiler alert number one, Bonferroni assumes that this thing is essentially 1 over n or 0. False discovery assumes this thing is somewhat large. That's one of the, and we'll talk about where all that comes in a second. So here's a simple way to do that, and I can see if I can pronounce my, I guess it's Norwegian, Schweder-Schwab-Platz. Help me out here, any native speakers? No, anyhow, here's the idea. So what I've done here is I've done 2 simulations, and this is a QQ plot, right? I'm plotting the rank over 1 minus the p-value, and the reason for that is that as this thing gets near 1, this p-value is tiny. So in this first plot up here, I have 100 tests, and each test is a random distribution. So as we did before, I take, let's say, a sample size of 50 drawn from the null, I compute a p-value. So over here, this straight line with the black triangles, all 100 of those are from the null. Now in the second one, what I did was I assumed 80 were from the null and 20 were from the alternative where it had an effect. And what you see is it looks nice and smooth, and then it kind of kinks up right here. So if you figure out where to cut off this line and continue it, that value there, n naught, here, which in this case is pretty close to 80, that's the number of nulls in your sample, just looking at your p-values. Pretty cool, huh? And how do you tell where this kink is? Something called the Chow test lets you do that. The Chow test basically says, fit a single regression to the data, compute its error sum of squares, fit a second regression to the data at your cutoff point, regression 1, error sum of squares, regression 2, error sum of squares, and look at the ratio of those independent errors over the total, and you can find out where that's maximized. And that's the Chow test in econometrics. It's a way to ask where you have cutoffs in these sort of things. And I will send Kavita chapter 10 to post on the Slack page. So that's one way to do it. Pretty cool, huh? Here's another way to do it. So here's real data. I think this is like something in pigs or whatever, from animal breeding. And here's a histogram of p-values. Now it's pretty clear that not all of these are from the null. That's a big excess. It's obviously a big excess. If you wonder how much of an excess, you can just simply do the binomial Laplace calculation on it. So how do I estimate from here what n naught is? We use a very simple idea from the uniform distribution. And that simple idea is, how many do I expect between A and B? For example, between 0.3 and 0.6. Well, a uniform just has a value of 1. The density function is just a flat value 1. So the fraction I expect, the number I expect between A and B is just simply the true number of nulls times B minus A. So I take the number of values in A and B and divide it by the A and B, and that gives me an estimate. I could also simply divide that by n and get a fraction. So on this data here, suppose I choose 0.4 to 0.6. When I do that, 0.4 to 0.6, when I count that, I have 154 tests. That distance is 0.3. I divide 154 by 0.3, and then what I get is 513. Now suppose I go from 0.3 up to 0.8. Same logic. Now in that I get 537 tests, and that, 322 significance if you count them up in here, tests in that bin, and there's my interval, and that estimate is 537. So you use different regions in there, you're going to get different estimates. And the idea is you try to avoid the end, certainly this end. You try to sample from the middle, but it's also clear the more you sample, the more precise your estimate is. Now, if I have an excess of 1s, which can happen if I have a one-tailed test and I'm actually way below that. If you don't have that, a common estimator is Story and Tibarashi's estimate, which is now I look at between some value lambda and 1. So what I do is I take, go back here, is I take this and I take from some value 0.3 up to 1, 0.4 up to 1, 0.5 up to 1, et cetera, and then do a model fitting and see which fits best. So you're obviously making some assumptions there, but it's not, it's one way to go. There's a better way to go, and that's your next new friend, the beta distribution. How many people have heard of the beta distribution before, besides Nick? It's not that uncommon, but let's have a little fun table of distributions over here. And just for fun, let me just kind of remind you, if U is a normal with 0 mean 0 and variance 1, then mu squared is a chi-square 1. If I sum up I equals 1 mu I to n, that's a chi-square n. If I compute chi-square k over k divided by chi-square m over m, so I'm summing k here. The reason I divide by k is the expected value of a chi-squared is degrees of freedom. This is an F distribution with k numerator and m denominator degrees of freedom. So those will come up a little bit later on. That's kind of the connections between those. When you see chi-square and F, et cetera, that's where they come from. Beta, so let's go a couple of distributions which are popular. So number one, I go to 0, infinity, and that's the gamma distribution. And the gamma has two parameters, a size and a shape parameter, and it either falls off or has a hump in the middle. Falls off, it's an exponential, hump in the middle. So that's used quite a bit. The other one is beta. Beta, again, is a two-parameter distribution, but now you're over 0 and 1. So frequency data is very, very often used. And beta is nice because it's really, really flexible. All of these 4 different curves are from a beta. So in a beta, if I take A equal B equal 1, I get a uniform. That's kind of cool, over 0, 1. If A here is less than 1, I get a big, strong peak at 0. If B here is less than 1, I get a big, strong peak at 1. I can also adjust these to get a hump in the middle. So it's a rather flexible distribution. I can get this U-shaped distribution as well. So if you think about fitting p-values, you might think about fitting a beta with a uniform, and then another beta, and because you can adjust the shape, you can, for example, have situations where you've got excess of p-values near 0, excess near 1, or a combination of both of those. And David Allison is the one that suggested that, and what he says was you take all your p-values and you fit 2 distributions. That means, in this case, 3 parameters. Parameter number 1, what fraction is from the null? Parameter number 2, 2 and 3 are the A and B as you fit the p-values that aren't from the null. So this is a way of, instead of eyeballing it, of actually estimating it. And here's a real example. So the boxes here are the histogram you see. The solid line is that fit of mixture of betas. And the way you fit this, by the way, is think about the p-value as a data point. So I've got 600 p-values. I can then use maximum likelihood to fit this model. Estimate the fraction, which are from the null, and then estimate the A and B shape parameters on that beta of those that aren't from the null. And that second one is flexible. I can have a big peak near 0, big peak near 1, a combination of those. Any deviation from the null, I can do that. So here I fit this, and this dotted line is the beta that's fitted from the uniform, and pi of all my observations are from that, and then 1 minus pi not are not from that, and the fit for that looks something like this. You put those together, and you've estimated it. So this is a clean way of estimating that fraction pi 0 of tests that are truly from the null hypothesis. So key points so far, if I have a distribution of p-values that are all from the null distribution, they're uniform. I can then use that to ask, do I have an excess of significant results in my set of tests? I can take my set of tests from the p-values and compute the fraction of all of those tests that are likely from the null. And I showed you a couple of different ways to do that. Let me just see where we are here. Yeah. So that was basically about, do I have an excess of significant results? And if I have that, is there things I can say about what fraction? And again, we're not saying which test is significant, is from the alternative. We're saying what fraction of all the tests are from the alternative, because 1 minus that is a fraction, sorry, we compute the fraction pi 0, which are from the null, because 1 minus that is the fraction of tests which do have an effect, which are from the alternative. So questions on basic stuff about p-values. Some pretty cool stuff there. And again, there's some details here. That's why you have the appendix. Just kind of try to get the basic idea and kind of get the understanding. If I've done nothing else, then make you think about when you have a bunch of p-values, maybe I should plot them out. Maybe I recall this weird guy gave some lecture in India where the food was good, and then go back and look in your notes and dig something out. So I view a lot of these lectures as throwing ideas out there which may not germinate right now, but as we say in the US, it's like a bad burrito. It may come back a lot later. So that's the idea about estimating the fraction of tests that are truly from the null hypothesis. Yeah.
So presumably the ease with which you can estimate this proportion of null will depend on the shape of this beta that you get. Here it seems like, I don't know, with this shape of beta, yeah, it can get...
So let's go back to beta.
So do we have expectation for how these deviations should look for? And
and the good news is you don't care about that because the model fits it for you. So for example, this U-shaped curve here could be the A and B parameters that you get from fitting your beta. And that would say in excess of 0 and excess of 1. For example, that would be a one-sided test where some are way below and some are way above. The way below would give you excess P, the way above would give you excess P near 1. So the actual shape of the p-values from the alternative, you don't care about, you just try to get the best fit.
But since the fraction of uniform would just lift everything up or down,
right.
I guess perhaps twisting the parameters a little bit would get you a little bit lower, and then you estimate lower P.
So the maximum likelihood does all of that stuff for you, right? And it gives you uncertainty on your proportion pi naught, the uncertainty on your A and B parameters. So the cool thing about maximum likelihood is that you just plug it in and it fits all of those for you. Because typically people don't care about the shape of A and B. What they typically care about is pi naught, what fraction are true nulls and what fraction are from the alternative. So with that background, let's move on to the next part, which basically has to do with controlling the family-wide error weight, also called the genome-wide error rate in genomics. And again, we saw the idea, I'm testing 30,000 transcripts. And I want to, obviously I can't use P of 0.05 because I'm going to testing each one at their difference between two tissues or two conditions, or the sexes or whatever comparison you're making. I've got to basically test each one at a smaller level if I want to have over all the transcripts I looked at, if I want to have the error rate over all those tests be 5%. So we call that controlling the family-wide error rate. And one way to think about why people are so anal about this originally is a lot of statistics came from drug testing. And in drug testing, you have your current overpriced, overhyped drug that just barely works, but you can make $1 billion off it here. Then you're testing thousands of other candidates. You're extremely conservative because you have something that works, i.e. your company owns a patent on it. And so you're extremely conservative. Now contrast that with the genomic study. Looking at 30,000 transcripts, how do you expect none of those to show any differences? Nobody. The question becomes what fraction do? And in that setting, the Bonferroni is not the right thing to do because in that second setting, our concern isn't paranoia over making a false positive. Our concern more is I have a large set of data, I want to distill it to a much more concentrated set of data where sure I'll make some error, but I want to control how many of those things I put in this set are actually not from the category of having no effect. And that's where false discovery comes in. We're working up to that. So the standard, again, here's a little overview. We'll talk about standard Bonferroni. Then we'll talk about cool things like sequential methods, Holmes, Symes, Hormels. We'll talk about correcting for the effective number of tests. Then we'll talk about false discovery. Then we'll talk about using a Bayesian framework. Again, I motivated that by whether or not something is significant once you declared it shouldn't then change as you add more tests. But under Bonferroni, it does. So the Bayesian framework is a nice way to think about that, and a lot of the human GWAS are moving to Bayesian. The classic paper in human GWAS is the WCCC, a Wellcome Trust Case Control Consortium. And in that, that was 2007. Everything people do in GWAS now was laid out in that paper. People just refined it a little bit. And in that, they talked about using a Bayesian method to control for false discovery. Then we'll talk about permutation tests and extreme value, and my favorite, the Trinity theorem, which is a great term. So here's the standard Bonferroni. If I let alpha, and again, I'll give examples of all of these in a second. It's a little bit abstract, but I'll make it concrete. Let alpha be the significance. So I'm going to declare a test to be significant when I compute its test statistic, and the p-value for that test statistic by the criteria I'm using is alpha or smaller. So what I want to do is, if I have n tests, and I want basically, so what is this term here? Well, alpha is the chance of a false positive. So 1 minus alpha is the probability that you don't have a false positive. What's the chance I get all n of them are not false positives? It's 1 minus alpha to the n. What's the chance I have one or more? It's 1 minus the chance of none. So this is the probability of having any false positive out of n when I test each one at alpha. So I can invert this. There you go. So you get the Dunsaka approach, which is basically this. But people typically use Bonferroni, which is the simple Taylor series, and these are essentially identical if n and pi are rather small. That's the classic Bonferroni thing you've all seen. You now see the trade-off. As you add more tests to control for that error rate of 5% overall, then each thing is tested with a smaller and smaller cutoff value as n gets larger and larger. So here's a hypothetical example. What I have here is 10 p-values, and we're going to see with this exact data what the difference in using these different methods are. So under strict, don't ignore the bottom row. Here, under strict Bonferroni, so I have 10 tests, and I want to test each one. I want the overall error rate to be 5% over all 10 tests. I've got to test each one at a 1/2 level. So here, probability here is less than a half percent significant. Here, less than a half, close but less than, genome family-wide significant. Here, too big. Boom, you're done. So you picked out 2 of those 10. Okay? Well, as we'll see, other methods will pick out more and pick out more in a legitimate fashion. So in Bonferroni, what you're doing is that this cutoff value is the same over all tests. Sequential Bonferroni, realize as you look at different tests that n actually changes. So the simple one is Holmes' method, and blah, blah, blah. It's really, really simple. So you test this one at 0.05 over 10. If it's significant, you now have 9 tests left. Test this one at 0.05 over 9, over 8, over 7, over 6, etc. And what you now see, by the way, over here, this is just simply testing it at 0.05 because you're over 1. So what you see now is with the sequential Bonferroni, instead of 1 and 2 being significant at that 5% level over the whole family, you don't have 1, 2, and 3. So you picked up a little bit more. You can do a lot better. So the Sims criteria says, now wait a minute, let's look at things backwards. So here, if it's not significant, we stop. If it is significant, we chunk along until we finally get into something where it's not significant. That's what the Holmes method does, the sequential Bonferroni. The Sims method says, wait a minute now, let's work backwards. Let's keep on going down until we get a result that is significant. Here's the family-wide value for one test. No, not significant, not significant, not significant, not significant. Hey, significant. So all of these are now significant. Pretty cool. So Bonferroni gives you 2, sequential Bonferroni gives you 3, and Sims gives you 6. Wait till you see what false discovery does. There's another method, it's a little more bookkeeping, it's called Homel's method. The example 6.4 does the bookkeeping. I could do it for you, it'd take about 5 minutes, it'd be boring. Just be aware there's another method, and this often also outperforms even the Sims method. But in this data set, you still get 6 like the Sims method. So that's pretty cool. You've got Bonferroni, but sequential Bonferroni says that once you examine a test, when you move on to the next test, you've now changed your N. Now this assumes independent tests, we'll talk about in a second. But these sequential Bonferroni methods are actually quite nice at picking out more things. We still typically don't use them because there are better ways like false discovery, but it's something to keep in your toolkit. If ever you're gonna do a Bonferroni, always do a sequential Bonferroni. So one issue we have is tests can be dependent. So now a little fun bonus stuff about the eigenstructure of correlation matrices. It's real quick. So let's imagine I have SNPs, and for the SNPs, I compute a correlation matrix C, which are all the pairwise, that is LDs. I've got ones on the diagonal, I've got a bunch of zeros, I've got little things on the off diagonal. Those are ones, not dashes, by the way. So fun fact number 1 from your matrix algebra course is the trace of the matrix C is simply the sum of the diagonal elements. So this is n by n because I have n tests. So what's the trace of this matrix? It's n. Fun fact number 2, the trace equals the sum of the eigenvalues. So what's the average eigenvalue for a correlation matrix? It's 1. Now we're going to use that because the idea is with these Sims method and Bonferroni methods, I'm assuming independent tests. So if tests aren't independent, I'm being overly strict. Suppose all the tests are completely correlated. Well, I can just test that as 1. If I test it as 8 or 10 separate tests, I'd lose power. So people have thought about using this correlation structure among your data. And again, we use a correlation matrix because it has these nice properties. And the first use of this was Jim Chevroot. And Jim basically said, well, we can think about 2 extreme scenarios. Number 1, all n tests are independent. If they're all independent, it means I just have zeros off here on the off diagonal. So I've got n eigenvalues and they're all equal to 1. Conversely, if all the tests are completely 100% correlated, then n minus 1 of the eigenvalues are 0 and 1 eigenvalue, again, these have to sum to n, they're all non-zero, that eigenvalue now, the leading eigenvalue is now n. So Jim looked at that and said, wait a minute now, so we can think about the variance in the eigenvalues of this matrix. If the eigenvalues are all pretty uniform, I probably have independent tests. As the variance gets bigger and bigger and bigger, as emphasized by here, if only one of the eigenvalues is non-zero, then the variance is n. So Jim proposed this little simple estimate of the effective number of tests based upon the variance of the eigenvalues of that correlation matrix. It's pretty straightforward to do. Patterson in human genetics basically independently came up with the same idea. So the idea is if you have data. Yeah, question, Nick?
Yeah, you apply this to a set of test statistics, which are called the associations.
Yeah, so we look at the so you can, what you do here is you look at the correlated data. You can project that in the test statistics.
But are there assumptions that these have to be assessed independently?
So there's no, if you assume the data are correlated, then the correlation for test 1, the correlated data carries over to test 2.
So are there any restrictions on?
There are, but they're a little bit technical. See the Chevroot and Patterson and G and Ling for those. But you get the basic idea, right? It's kind of cool. So then Lee and G came along and says, well, we can do a little bit better than that. And their logic was, well, suppose that I have C identical tests. So that will give me an eigenvalue that's bigger than 1. However, if tests are only partially correlated, remember the eigenvalues have to sum to n. So if some of them are much bigger than 1, then some have to be less than 1. So they said, well, you know, if tests are partly correlated, then they give eigenvalues less than 1, and I can use that information. So here's an example. Here's an eigensequence. Eigenvalue 4.1, 3.5, 1, 0.5, 0.1. And what Li and Xi said is you look at this data in two pieces. Number one, count the number of eigenvalues that are one or more. That's 1, 2, 3. So I've got 3 independent tests here. Part number 2, take what's called the floor. So here I've got 0.1, 0.5, 0, 0.51, and add those up. These differences here on the end are due to partial dependencies. That now adds 1.2. So now I have effectively 4 tests. 1 for here, 1 for there, 1 for there, and then all the residuals give you basically 4. This is called the Li-Xi method. Pretty cool. Again, these are all approximate. If you have real data, compute all of these methods on them and look and see that. Typically, Chevrolet is a little bit too conservative. It gives you an any value that's too large. Remember, what you want to do is make any smaller. That makes your adjustment in Bonferroni smaller. And Li and Xi is something that typically performs a little bit better than Chevrolet. And there are others in the references you can are given in that. So that's the idea basically about getting an effective number of tests. So what do we have so far? We have Bonferroni, sequential Bonferroni methods, notably Holmes and Sims approach, typically Sims. What you do is, in sequential is once you do a test, then you change the number on the bottom because that test tells you something. Then we talked about all those methods assume independence. What if you don't have independence, which is very often the case in SNPs? Then you can use the correlation structure among those SNPs to estimate what that effective number of tests is. That's kind of what NE is, an effective number of tests. You may have 1 million SNPs, there's only 1 test, or there could be 5,000 tests, or it could be 1 million tests. And these eigenvalue methods give you a way of getting at that. So that's kind of classic Bonferroni, sequential Bonferroni theory. Questions or comments on that? Again, I want to stress all this stuff is in the notes, Appendix 6. I would highly recommend that you read Appendix 6 in the next week or so, because once you've seen this, then it'll be a little bit easier to read and go through. Nick?
I'm disturbed by your saying that you should try all the methods and see which one you like.
I didn't talk about p-value mining. When I say try all the methods, look for consistency, not the best. So what Nick is talking about is the insidious practice of p-value mining, which is I'll just keep trying methods till I get something significant. Don't endorse that. Although you could put that in a meta-analysis. I don't endorse it. What I do endorse is if you have your data, do several different methods of analysis on there. If all the methods give you consistent results, you're fine. If they give you inconsistent results, you got to look more carefully at your data, see what's going on. Maybe you didn't adjust for lack of independence among tests or something like that. Other questions before we talk about FDR? Yeah.
I have a
Not
question.
Franklin Delano Roosevelt, by the way.
I'm not sure it's going to be easy to answer it, and maybe it's just
Okay.
part of my homework, but you indicated that the eigenvalues would tell us about the partial correlation between the traits, and I was wondering whether you could expand a little bit on
Yeah,
this
so,
that I would have...
it's more kind of a verbal argument. Li and Xi, put some jargon there to make you think it's like hardcore, approved by mathematicians. But the idea is you can think about the eigenvalues having 2 parts. If I have correlated tests, let's imagine they're perfectly correlated. I've got 5 perfectly correlated tests. That's going to give me one eigenvalue of value 5. So I count that as one test. So I go and count all my eigenvalues and count all those which are 1 or more. Don't count their value, just count. So 4 counts as one test, 3 counts as the second test, etc. Then I've got some residual part. It's not exactly 3, it's 3 point something. It's not exactly 0, it's point something. It's not exactly 1, it's 1 point something. So Li and Xi says, well, then take all those fractions away from the integer and add all those up, and those are partial correlations, which then give you additional tests which are in there. The idea is, if I have an eigenvalue 4, I probably have 4 completely dependent tests. If I have eigenvalue 3.6, well, that's one test for 3, and that 0.6 means I probably have some combination of tests that are correlated. We're going to call that value 0.6. So a little bit of an ad hoc about that. Read the Li and Xi paper, but that's kind of their basic idea. And again, these things aren't mathematically rigorous, but they're ways people look at to try to kind of improve the fit. So you'll see, for example, a lot of the gene-based GWAS tests use SIMS, where they take sims and instead of dividing by n, they divide by n sub e using one of these different methods. So these are kind of the pieces that get put together. And again, don't think too hard about the details. Get a feel, and then the next week or so, go back and read the notes. What I highly recommend is print out the notes and then write comments on them. I've got big margins for that. And then just put that away, or even better, put a PDF of those notes in a file, and then put a copy of your notes, maybe make a short Word document, and put that in a file. So whenever I teach a course, I always recommend organize it within a day or within 2 or 3 days after you're done. Put all the PDFs in folders, and then any notes you have, unless you've got like 4 binders full, any notes you have, maybe make a short Word file about some of the key points, and then put them in there. Then you'll have a reference to come back to 4 or 5 or 10 years from now. So just good organizational tips. Let's now get into the false discovery rate, the FDR. So Bonferroni essentially assumes that all your tests are from the null. That is, again, null means they have no effect, H naught. Alternative means they have some effect. How much, we don't know. And that's the power issue. If the effect isn't much power, it's hard to detect them, but they're not from the null. So in modern genomics, we expect a subset. So pi 0 here is assumed to be essentially 1 under Bonferroni. Under here, pi 0, the fraction of tests which are truly from the null, may be something like 0.9 or 0.8, maybe 0.72. When you do that, it changes things. In such settings, we'd like to control the false positive or the false discovery rate among those tests we declare to be significant. This is a subtle shift, and I'll talk about it a lot and give you examples. So suppose all of you are statistical tests, and only this front row I declared significant by whatever criteria I use. So I have 8 tests in there. Suppose that 4 of those 8 tests are from the null. These 8 tests which are significant are discoveries. 4 of those 8 are false discovery, so the false discovery rate would be 4 divided by 8. If one of those tests is from the null, the false discovery rate is 1 divided by 8. So it's a natural way in genomic data, where what we're doing in genomic data is we're taking a large number of things we're testing, like transcripts. We want to distill that to a smaller set for further analysis. We want to be careful about that set. We want it to be highly enriched. We don't want to be so anal about it that nothing is in there. Bonferroni, very little is in there. With false discovery, I now have 4,000 genes, and there's only a 2% chance that any one of those genes that I've declared to be significant is a false discovery. That's why false discovery rate is more widely used in the genomics era, because it encapsulates the fact that you expect some of your tests to be from the alternative. Bonferroni, you're assuming basically that none of them are. And again, I'll show those connections as we move along. So a false discovery rate of 1% means among all those tests, for example, you have 1,000 tests that you declare by some criteria to be significant. Among those 1,000 tests, only 1% or 10 are false discoveries. So it makes sense to focus on that set of 1,000. It's much larger than it would be if you use Bonferroni, and yet the number in there which are false is small, so you have a distilled concentrated set for further analysis. That's why a lot of people use FDR. So how do we get to FDR? Here's kind of a little table. So called significant, not called significant. These are truly from the null. These are from the alternative. So this is the total that are declared to be significant. So the false discovery rate is F divided by S. To give you some perspective, what is the false positive rate? The false positive rate says, if I pick a random test from the null, what's the chance that I'm going to declare it to be true? So a random test from the null, that's the false positive rate. The false discovery rate is I condition on it being significant. Subtle point, I'll mention it several times. Again, that's why it's good to hear this and go back and look through the notes. So this is the false discovery rate. What is the false positive rate? Well, N naught is the number of tests that are truly from the null, and F of those are significant, so F divided by N naught. So you see the difference, F divided by N naught. I'm conditioning on you being from the null. Here, F divided by S, I'm conditioning on you being significant. F divided by S is false discovery, conditioned on being significant, conditioned on it being from the null. F divided by N is your false positive or rate. And again, I'll work through a bunch of examples of that. Another way to do that, let's actually work out and make this a little bit less messy. The cool thing about false discovery rate is it adapts to your current data set. So the nice little program to do this for you very quickly. So to set an FDR, I want to find the critical p-value for each comparison. So suppose I set that critical p-value as tau, for example, 0.1%. We'll just call it tau. If I have n tests, then I expect n times tau to be the expected number of false positives. You can improve that if you have an estimate of n naught. This would be the expected number of false positives. So what is the FDR? It's really simple. How many of your tests have a p-value less than tau? That depends upon your sample. It's different for every sample. That's what's on the bottom. On the top is the expected number of false discoveries for that tau. So suppose I have 1,000 tests and I set that critical value at 1%. What do I expect? I expect 10 false positives. Well, how many discoveries do I see? Well, that's data set specific. So suppose for that critical value, I look at those 1,000 tests and I see, oh, hey, 120 have a p-value that's 1% or less. So my FDR is the number of tests that are significant in my sample. Out of the 1,000, 120 are for that t-value, and then that's on the bottom. On the top is the expected number of false discoveries, so my false discovery rate here is 8%. Now let's imagine, instead of going 1%, I take 0.5%, so I expect 5 tests to be false positives. Again, I have to go back to my data. Again, it's different for every data set. And now out of that 1,000, instead of 120 having a p-value of 1% or less, only 110 have a p-value of 0.5% or less. Now my FDR is 5 divided by 110, or 0.45. So what you see is you take your data, and a program does this for you, and you change that tau value. N times tau gives you the number of false discoveries, and then with that tau value, your data set gives you the number that are significant, and you move that up or down until you get the false discovery rate you want. So if I set the critical value of testing each of these 1,000 tests at 0.5%, that gives me a false discovery rate of about 4.5%. So think about that basic idea. As you change that cutoff, as that cutoff gets smaller, the number of false discoveries gets smaller, but then so does the number of significant tests. So a thing on the top you always know, and again, you can improve it if you can replace n by n naught by some of those methods we talked about. That thing on the top stays the same for a given tau, but on the bottom depends upon your particular data set. So false discovery rate is a very simple thing to apply operationally, and again, there are programs that do this for you very, very quickly. One more thing, and then I'll see if there are questions. So again, it depends upon who you condition on. So the false positive rate is, given the test is null, that is, it's truly null, what's the chance I declare it to be significant? So I'm conditioning on it being null. The false discovery rate is, hey, I have a test that's significant. What's the chance that it's from the null? So you notice these are kind of flipped, right? Declared significant, I'm conditioning, I'm flipping the conditional. And so here, this is something I typically don't know. I don't know which ones are from the null, but here, I do know which numbers are declared to be significant. So they often sound really similar. And again, that's why I recommend reading Appendix 6 after this, because now that you've thought about these ideas have been perturbing you a little bit, it's easier to go through and read and pick out some of these differences on there. Okay? So let's see where we're next here. Yeah, so this data set we had before, let's go, remember, so Bonferroni gave me 1 and 2, Holmes' method gave me 1 and 3, Sims gave me 1 through 6. What does FDR give me? 1 through 8. So among these 8 tests, there's only a 5% chance of having a false discovery. So that's what people like, and think about that as a much larger scale. So people like false discoveries.
So, but usually you don't do it stepwise, first Bonferroni and then FDR?
So you can set your p-value however you want, right? You can set your criteria for testing however you want. Then you can go back and do this. But typically, when people do FDR, and I'll show the connection in a second, when people do FDR, they're getting a p-value for a test, and then they're asking if it's significant. That's significant. When you get the significant criteria, that's where you might use Bonferroni or sequential Bonferroni or something like that. Or just don't use it at all. Just give a raw p-value, and then use that. A raw p-value is probably easier because it's a little bit easier to compute the number of false discoveries in that situation. And again, I'll show how all these things are connected in a second. Don't you like it how I keep on promising I'll get there? We'll run out of time. So I would have told you we were out of time. So the connection, very influential paper by Newton Morton, who's a well-known name, kind of the founder of human linkage analysis. This paper is older than I am, which is actually saying a lot. 1 point in that distribution. This is called the posterior error rate or the false positive reporting rate. And now, what are we asking? We're saying, given I pick a random test that's significant, condition is significant, what's the chance it's a false discovery? When you look at this, it gives you a lot of connections between what's going on with Bonferroni and false discovery. So we can do that. We can use Bayes' theorem. Bayes' theorem allows you to flip the conditional. And again, you can read about that in the notes. Then you compute what this is, and what this is quite simple. So alpha is the significance I'm testing at. Beta is a type 2 error. Beta means that a beta of 30% means that when my effect is indeed not 0, 30% of the time I won't have enough power to distinguish it, that is, call it significant. So 1 minus beta is power, and notice what comes in this posterior error rate. What comes in critically, and again, I'll give examples to make this a bit more clear, is this pi naught, the fraction of tests which are truly null. So what you can show is, you can turn this around and say, okay, if I want a posterior error rate of 2%, that means of all the significant tests I look at, only 2% are false discovery. So here's how you can get that. You've got to know power. This allows you to set the A, but look what comes in here, pi naught. In particular, if you assume that only one of the things you're testing is from the null, so pi naught is 1 over 1 minus n, what does alpha become? That's Bonferroni. So if you assume that all of your tests but one are from the null, that is, essentially none of the things you're testing you have any faith is going to be from anything other than the null, that's where you use Bonferroni. Conversely, if you expect some are actually from the alternative, that's when you use false discovery. Let me make that a little bit clearer with a couple of examples. So what Morton did, this is really for human linkage, and he said, well, I've got 23 pairs of human chromosomes, he was the first one that said 23andMe. So 1 over 23, 5%, is the prior probability of linkage. If I pick 2 random loci from the entire genome, again, rough approximation, chromosome different size, I expect there to be about a 1 in 23, about a 5% chance that by chance are on the same chromosome. So pi naught is 0.95. So 95% of all random comparisons you make are unlinked. Suppose you have a type 1 error rate of 5% and you have 80% power to detect linkage. If you plug in the result here, the posterior error rate is 0.54. It means 54% of all things that you declare to be significant are false positives. So that's why it's called the posterior error rate. If I just simply take a random significant test, with these numbers, in particular, 95% are from the null, there's a 54% chance I declare it to falsely. How does that come up? If I draw 1,000 pair of loci, 950 are expected to be unlinked. 5% is a chance I call any one of them significant, so I expect about 48 of those show a false positive. If I have 95%, so 50 are expected to be linked, and of those 50, 80% of the time I can declare them to be significant, so I have 40, so my error rate here is 47.5 divided by these two, or 0.54. What value of alpha do I need to control to make that error rate of 5%? So if I pick a random significant test, Again, significant test, not a null test, a significant test. If I pick a random significant test, hold that question. I think the next slide often answers this, unless it's a clarification question. If I pick a random significant test, 54% chance with this alpha, it's a false positive. If I now pick 0.0022, now it's 5%. One more slide and then open for questions. I think this kind of ties things together. So what are the implications of that for multiple traits? Well, let's do this. I set alpha equal 0.5%. I assume the power is essentially 1. And I have 5,000 tests, but I have 2 different settings, 2 different genes, 2 different traits. For example, body mass and schizophrenia. Suppose that in trait 1, the alternative is very rare. So pi naught is only 1 out of 5,000. So pi naught is 0.9992. Under this setting, I expect 25 false positive, 1 true positive. The posterior error rate is 96%. Take the same power, the same alpha, and now assume that 500 of those 5,000 tests are from the alternative. Pi naught now goes to 0.9. Now my posterior error rate is 0.4%. Why the big difference? Over here, almost everything you're testing is from the null. So you're going to get false positives. Over here, you have a smaller number you're testing from the null, and you have a larger number of ones you're going to get true positives on. So this parameter pi naught is really critical. If pi naught is essentially 1, all the tests are from the null, that's where Bonferroni comes in. If pi naught is 0.9, then you get big differences by putting that in your model. And with that, your question.
My question feels very silly. It's on the previous slides.
Previous slide, yep.
You get 87.5%, and that lost me.
So right here, so
Just below 47.5 divided by 87.5.
right,
Where is the 87 going from? You sum these two, is that right?
You sum those two.
You sum these two.
Yeah. So 40. So how many tests are significant? That's the expected number under false positives, and that's the number given 50 are true and power is 80%. That's the number over here. So that's the number of false positives. That's the number of true positives. The reason this isn't 50 is my power is not complete. If this was 1, then I'd get 50 of those. This would now become 47.5 over 97.5. So you're kind of seeing the connections. One of the connections are Bonferroni basically assumes that essentially nothing is from the null. Whereas if pi is different from 1, maybe it's only 0.8 or 0.9, that's what you want to then adjust for. Yeah, question.
Can you give another example on how you'd use the PR?
So the posterior error rate is the false discovery rate. It's just, so what you're doing is you're conditioning on the test being significant. And the posterior error rate, you said I pick a random test. And if I have a random test, 3 parameters tell me what that error is going to be. Number 1, the significant test I'm doing, because that'll tell me what fraction of false discoveries. Number 2, pi naught, which is the fraction of all the tests that are from the null. And number 3, beta, the power. So you want to put beta equals power of 1, beta equals 0, then you can just look at what really drives it is alpha times pi naught. So if pi naught is different from 0, that is 90% instead of 1, then when you do, if basically almost none of your tests are from the alternative, they're almost all from the null, a significant test is a high chance of being a false discovery. If you have a fair number of things which are from the alternative, a significant chance has a fair chance of being a true discovery. So false discovery rate goes down. That's that connection between pi naught, FER, and Bonferroni. Again, a lot of stuff going on here. It's kind of hard to keep some of these conditionings straight. It's late in the day. So I'm going you to kind of think about these things, then go back and read through the Appendix 6. Any other questions on false discovery before we move on to Bayesian approaches? So I've kind of teed up Bayesian before. By any of these methods, your significance or FDR depends upon the number of tests. But it makes no sense that if you have done a number of tests and declared a thing to be significant, if you then add new tests, ones you declared to be significant before are no longer potentially significant. Why? Because that alpha over n gets a lot smaller. That kind of logically doesn't make sense, but it makes sense the framework of Bonferroni. Is there a way around that? And the way around that is Bayesian methods. We haven't said a lot about Bayes in this course. The Bayesian framework is thinking about, so everyone is a Bayesian, right? Let me give you an example of everyone's a Bayesian. If I pick a random person here and ask him a question about diffusion theory, well, the odds are kind of small. If I pick Nick, I have prior information. I know he certainly doesn't know it, right? So we all are Bayesians. You meet someone, you like them, then they do something bad to you, and you readjust your prior. So you have some prior belief, you have an interaction, then you have a posterior belief. So Bayesian is simply that, using prior information, taking some expectation, and then readjusting it. And there's gazillions of things. There's a great book called The Theory That Wouldn't Die. It talks about Bayesian. It's kind of a, if you have a long plane ride back, download that on your Kindle. So here is the idea. And again, the Wellcome Trust Case Control Consortium said this. It was probably Matt Stevens and Balding. It is not the number of tests performed, but rather the prior credibility of the hypothesis that is important in interpreting a set of observed associations. That is, if the hypothesis is unlikely a priori before the data to be true, we should require strong evidence to be convinced of the truth. For example, 1976, Mariner, the Viking probes landed on Mars. One of their experiments showed an increase in assimilated labeled carbon. Well, if your prior probability of life was pretty high, you'd say that's evidence of life. If your prior probability is really low, you go, well, that's intriguing, but it's not strong enough. So that's the Bayesian framework. It's kind of abstract, but I'll give a very concrete example in a slide or two. So here's the idea. So this is a conditional probability. I can replace it. So look at Bayesian flips these conditioning on tau. This is my test statistic, being that this is under the chance of getting that extreme test statistic, under the alternative and under the null. I can put together and get this nice expression. 1 minus beta is the power. And again, I'll give a real example with numbers. Alpha is the significance. And now this ratio, this posterior odds ratio of it being true divided by the chance of being false, now that depends upon the prior odds ratio. This is if I pick a random SNP, the probability it has an effect divided by the chance a random SNP doesn't have an effect. So this is kind of abstract. Let's make it concrete. You can write something called the posterior probability association using that. But here's, again, make it concrete here. So here's an example. So 2 scenarios. Scenario number 1, in both cases, I'm testing 1 million different linkage blocks. So I got a really chopped up chromosome. Scenario number 1, I expect 10 regions out of those millions on average to influence the trait of interest. And suppose I have a 50% power to detect each of those. I want an odds ratio of 10 to 1. If I declare this here, I want there to be a 10 to 1. That is, you put $1 down, you get $10 back. That's a pretty good bet, right? What is that? What do I have to set this alpha value? We have to set it to 5 times 10 to the minus 7. You plug those numbers in. Of this, I have 5 true discoveries and 0.5 false discoveries. I get an FDR of about 9%. And I can check other value. Now let's take that exact same scenario, 1 million SNPs, same power. Now let's imagine that instead of there being 10 blocks, there's 1,000 blocks. So I've got like height. So now, what does that alpha value have to be to get an odds ratio of 10? Instead of 10 to the minus 7, then 10 to the minus 5. Why? There's a higher a priori chance that any random site is going to involve the trait. So because my a priori chance is higher, I need less strong evidence. If my a priori odds are really low, I need stronger evidence. So this Bayesian framework then, you notice what's not in here? What's not in here is n. N appears nowhere in here. Number of tests doesn't come up in here. And you can also relate it to false discoveries. So a lot of people are now using this kind of Bayesian framework because it takes you away from it. Now, it's a little bit complicated because you've got to calculate these complicated things called Bayes ratios. And by the way, notice this is from chapter 20, which you also have. So just look at 20.7b and you can get the reading on that, how you actually compute these sort of things. But the idea is it's a little bit of shift. The shift is I have some prior belief, and then I look at the data and I say, given my prior belief, I need really strong evidence if my prior belief is small for a random SNP. I need less strong evidence if my prior belief is modest for a given SNP. And you can use that then to adjust that cutoff value you're going to use. Yeah.
But so that belief is exactly what? Prior studies? So you would use it in meta-analysis or?
Yeah, so prior studies or just, what a comparable trait might be. And again, what you can always do is you can always ask, what's the effect of changing that number? And that prior belief, you think about that prior belief as a pi naught, right? So your ratio is 1 minus pi naught divided by pi naught. That's that ratio beforehand. So you don't even need numbers, simply what fraction of tests you think are from the null, or what fraction of your potential test, that whole universe of tests. The nice thing about this is that if your prior beliefs in that first 500,000 SNPs were significant, those prior beliefs don't change as you add more SNPs. Now, you could do adaptive Bayes, is as you add more SNPs, you realize, oh, my pi zero value was overestimated. So you can do that way, but you don't necessarily have to. So the thing about this is basically independent of the number of tests you do. Yeah, question.
I'm thinking in the concept of, in the context of GWAS, we often have linked SNPs. And so how does that come into this Bayesian? Like if the neighboring SNP is significant, my chance then...
Yeah, so there's a bunch of ways to handle that. Number one is you can trim all the SNPs but one. Number two is you can test each SNP. Yeah, they're linked, but that's okay. And you can use that linked information, and chapter 21 talks about that. You can use that linkage information to get a higher probability of where the actual causal site is. So people often trim it, just compute a p-value for each one. Sure, the p-value is going to be correlated, or the odds ratios in this case are going to be correlated, but that's okay. Now we've dazzled you with Bayesian methods, per popular demand, permutation tests. Kind of the gold standard. Anybody know who came up with the idea of permutation tests? When in doubt, say Fisher, right? In statistics, you're not likely off. That's a strong prior, right? Fisher. Although Fisher did call Bayesian methods the sin of inverse probability. But that's okay. So the idea is really simple. This is the gold standard. So George and Churchill and George had a couple of papers early on in Q-tail mapping. And the idea is your data sets are enormously complex. So each of you is an individual life measure. Each of you, I have height, or maybe I've got two variables, height and body mass. And then for you, I also have in that vector of data, I have a whole bunch of markers. In the early, Q-tail mapping with linkage, maybe a couple hundred. In a GWAS, it could be several thousand. So each individual point, I've got data on phenotypes and data on markers. Permutation test says if the data consists of this, for individual I have of either one or a vector zi of trait values, then for that individual, I have a vector mi of marker data. And I run a test and I get, test statistics. How do I tell when they're significant? Very complicated, lots of correlations. Well, it's pretty easy. You think about a deck of cards. In the deck of cards, you have a box with two cards in it. Card number one is that list of the phenotypes, which could be correlated. Card number two is that much longer list of markers. You don't change those cards, but you shuffle the top cards over the bottom cards. That is, you randomize or permute them. That breaks down any association that might be there in individual I between the markers and the trait values. Then you run your analysis and compute the highest test statistic or the highest on a chromosome, whatever you want. Then do that again, several thousand times. And suppose over the entire data, let's imagine looking at chromosome 2, over the entire data, I never get above a threshold of 3 more than 5% of the time. Well, that threshold you see is your 5% error rate for that entire chromosome. So really slick, conceptually straightforward thing to do. That's why people like it. Now, there's some issues with it. Issue number one is, what is the exchangeable unit? The example I gave you with marker data is obvious. You don't break up the phenotypes because they're correlated, you keep them intact, but you shuffle over markers. But now suppose that Nick and I are brothers. You can't shuffle us because there's other correlations in there that could be important that we don't incorporate in there. So we call this the exchangeable unit. What can I shuffle and keep the important correlation structure intact? Because if I shuffle Nick and I because we're family members, I break up the correlation from families, and that correlation could impact your test statistic. So one issue is, what are the exchangeable units? It's very robust when done correctly. The cons are, again, exchangeable units may be unclear. The other thing is, suppose you have several different studies and you're trying to combine them. So when you're combining them, so let's imagine you're testing several different gene sets. You test 1,000 different gene sets. Now you're trying to combine those genes into one p-value. Well, you run a permutation test on each. Maybe they're done in separate labs, so you can't combine the data. So in order to get a p-value of 10, you need to look at least 10 divided by p runs. So for example, if the p-value I need on each of the tests when I combine them, I get a reasonable overall rate, I may have to run 10 million permutations for every single different gene I'm looking at. That takes permutation tests to make them a little bit more difficult to apply. Chapter 21 talks about ways to get around this, but here's an interesting way, and this gets back to extreme value theory. It's a very cool result due to Fisher. Specifically, the Trinity theorem, Fisher, Tippett, and Genetico, basically says that if I'm taking draws from a distribution, and I want to ask, what's the distribution of the most extreme value? Not all the values, not one value. So the normal, you could tell how large any one value is going to be. If I pick 30 and ask what the extreme value there is, what's that distribution look like? Well, the Trinity theorem is super cool. It says, if you look close enough to the boundary of the tail, it's one of three things, and you're done. All continuous behaved distributions fall into one of three categories. If it's bounded, it's the Weibull distribution. And then depending upon this parameter kappa, you either get a Fichette distribution, which is heavier than an exponential, or a Gumbel distribution. The key is, this is a two-parameter family, kappa and tau, and you can estimate that from your data with maximum likelihood. So here's one way to look at this. It's often used, as used in chapter 27 for extreme walks. Here's the highest fitness. Okay, I sample and sample and sample and sample. When do I get the value higher than that? Sample and sample and sample. And so this distribution has one of three forms in the extreme tail. If it's bounded, it's Weibull. Weibull can go all over the place. Otherwise, it's either got a kappa of 0 or a kappa greater than 0. This is an exponential. This is what normal data falls like. Or if it's heavier, it's a Fichet. And the idea with using this extreme value theorem is you run your permutation tests, maybe 10,000 to 20,000. That's a lot, but it's a lot less than 10 million. You then use the extreme values in that to then use likelihood and fit, by the way, this is called the generalized Pareto distribution, this family here, depending upon what kappa and tau is. So you simply fit that with maximum likelihood. And again, since you're not bounded, you simply estimate kappa. Is kappa 0 or is it bigger than 0? And then that allows you to analytically look what the tail is going to be. Even though you sampled only 10,000, you can then analytically extend that tail out and look at what the cutoff value for 10 to the minus 5 or 10 to the minus 7 or 10 to the minus 8 could be. So this is a way of combining the nice approach of permutation with the idea basically of extreme value theorem for sampling in those tails. So kind of cool. Again, a little abstract, but keep it in mind because it could be useful. Yeah. 10, 20,000 times. And we'll do the whole permutation. And then keep track of those extreme values. And then take that distribution and fit it with the maximum likelihood. Because it's going to fit a generalized Pareto. Basically, it's going to have a kappa, of 0 or a kappa bigger than 0. Once you fit that, you can then extrapolate. Once you fit that distribution, you can then analytically compute what that cutoff value is going to be. Okay. Well, that's the exchangeability part. So I'm assuming you've matched the exchangeability conditions. That's a separate issue entirely.
Well, it's very rarely tested.
Yes, and it should be. There are things.
Why people think I can apply it always.
Yeah, so the things where it's good, but the exchangeability units really is kind of a critical issue. Okay. I'm probably going to run about 10 minutes over. So what have we looked at so far? Computing the probability pi of the number out of n of all the tests that are from the null. 1 minus pi is the tests that are true effects. We then talked about different ways of controlling for the p-value over your sample. We talked about Bonferroni, we talked about false discovery. We've talked about Bayesian methods, looking at odds ratios. Then we talked about another way to get it, which handles all sorts of independence, is permutation tests and how you can handle that. Now we're going to turn the question around. And now let's imagine I have a bunch of p-values, but my issue here is not to figure out which ones are significant by some criteria, but rather to combine them and get an overall summary for all that individuals. And the way to think about it is I have a set of SNPs in a gene. I'm trying to test if there's a gene effect. And one reason this might be useful, if you have diseases which are rare and have several different populations, in that gene, different SNPs may impact the disease in different individuals. So if you test any one by itself, you may not get enough signal. If you test them all and compute an overall joint p probability, you can often get a signal. These are called combining p-value approaches. And the original method was due to Fisher. So Fisher's idea, again, under the null, p-values follow a uniform distribution. So the log minus log of the p-value is a chi-square with 2 degrees of freedom. So let's look at this data set here. I've got 5 SNPs in the gene. None of those are significant. But man, that's sure non-random. It's really crunched up, right? That's not a uniform. They're all, think about it, right? These 4 are all crunched up. It shouldn't be that way. So it's clear they're highly non-random. But what's the overall significance value on that? Well, this is Fisher's combined test. So you take the sum minus twice the sum of all the log of the p-values, and if there's k of them, that follows the chi-square with k degrees of freedom. There are extensions, gamma methods, references on here. So for example, let's look at these p-values, 0.1, 0.06, 0.15, 0.08, 0.07. They're not significant. Man, they're all less than 15%. That's non-randomly clustered. So go ahead, take log, compute that sum. Here's what you get. And there are 5 tests, so 10 degrees of freedom. The probability that a chi-square with 10 degrees of freedom is bigger than 24 is 0.6%. So individually, none of those tests is significant, but they're non-random. They're clustered near 0. So when you look at all those together, you get a significant result, and there's your combined p probability. To show you how this is, another way this is kind of cool, is suppose you have 2 tests, each of which have a probability of 0.06. So they're not significant. And I have a test with probability of 0.05. Well, if you can use the Fisher's value here, the probability of 2 tests of 0.06 is 0.02. So 2 tests of 0.06 is really more power than 1 test of 0.05. Why? Because they're both non-random, shoved near the 0. So Fisher's method, you assume p-values are independent combined. These references here talk about how you handle correlated structure, kind of similar to what we talked about before with effective eigenvalues and things like that. That's one approach. Stouffer's z-score is another approach. By the way, probably the most obscure origins of statistics. In the after World War II, Stouffer was commissioned by, I think, the War Department to write a report on, the status of returning soldiers. And in a little appendix, in a footnote, this method is developed. And the idea here is that each p-value, I back transform that and get a z-value. So for example, if the p-value is 0, that's if the p-value is 50%, that's a z-value of 0. So I then basically take the sum of those, divide by the square root of the number of tests, And then I can take that score, then I can contrast that to what it would be under a normal. You can also weight that. So here what we're doing is we're taking data and we're transforming those p-values into normals, summing them up, then asking, comparing that to what it should be. So again, examples always help. So suppose I take the same data that I just showed you, I go ahead and then compute the z-values, standardize that, and what's the chance that a uniform normal is bigger than 3? It's about 0.1%. So Stouffer's value gives me a highly significant result. And people often report both Stouffer's value and the Fisher's value. Those are the two you most commonly see. There are others. Tippett's value is kind of cool. So remember we had before, if I test each with alpha, this is the overall level of significance. Well, let's just flip it around. This is my overall level of significance on the tests. So I plug in the smallest p-value. So n times the smallest p-value is my overall significance on the test. This is called Tippett's value. Pretty clean approach. Sims method is a similar sort of thing where, again, this is computing Sims, you kind of reverse it. You can get a p-value for that. And in all these methods, you can replace n by ne to a pretty good approximation. So again, these are different methods you look up. Don't worry now about what they are. Be aware that these methods for combining p-values, and you now have a reference to go where you can look at some of these things all together in one place. The new contender are Cauchys. So Cauchy, here's the Cauchy distribution. It's a two-parameter distribution. We won't worry about that. It's a ratio of two normals. You can think about it as a chi-square with 1 degree of freedom. And it's typically taught in probability courses because the area under the curve is 1, but n times that area, the tails don't fall off quickly enough, it's infinite. So it's a probability with no finite moments. But the reason it's really cool, and you can relate it to arctans, et cetera, I won't go through the details. The reason it's real cool is this fascinating result here. And that is that if I take a bunch of Cauchys and weight them and add them together, I get a unit Cauchy back out again. So I can have all sorts of dependency structure in the data, and yet I still get back a Cauchy. So people are now using this approach. Again, it's late, I'm not going to drive down into it, but just here's the idea. If I expect a lot of correlations among my data, I can use this Cauchy approach. I take a p-value, I then transform that p-value here into an appropriate Cauchy value. Actually, go here. Transform it. So this is the value corresponding to that p-value. I then sum those up, maybe a weighted sum. Then I basically then back transform that. This is my test statistic. This is a Cauchy. I can then back transform that and get a p-value out of that. So this is an approach people are using now because it's nice in that it handles arbitrary independence. So it's really a cool idea. Now, how robust it is not clear, but it's a lot more robust than I initially thought, just looking at it. Because there is some theory underlying it. You can take a weighted sum of Cauchys and get out a unit Cauchy. This is why the Cauchy never follows the central limit theorem, right? You can't sum up Cauchys and have that be a normal. So this Cauchy tail is a way that people use. Again, think of Stouffer's, but instead of using a normal, we use this odd distribution called the Cauchy. That's the end of p-value combining. Just have maybe about probably 9 more slides. People okay? No? Yes? Everyone else is left, right? Yeah, question.
Is there any experimental examination of when you try to optimize the threshold, whether the biology follows? Like,
yeah, you can. So I didn't. Chapter 21 talks about a huge amount of stuff. So, for example, you can weigh different tests based upon the functional significance. It's a region. It's a protein coding variant. It has a high predictability of having a misfold. There are all sorts of ways you can put genomic data into these the weights. Chapter 21 talks about that. I'm just trying to give you an overview. But you can certainly add biology to these weights.
I think, I'm not sure, I think my question was whether just optimizing the p-value threshold, whether somebody has looked at whether it really improved, whether the fraction of false positive was correct biologically.
Yeah. So people have looked at that with simulations. The problem is it's only as good as your assumptions. So you can simulate it. How you know it's good? Well, you know, under these assumptions, you can tell what you get. If you take real data, it's hard to check the assumptions on that. So if you look in the data literature on like gene-based tests, people now over the last couple of years are using these aggregating Cauchy methods. Again, the idea is that, so a classic line by Box, one of my favorite statisticians, he said, it's far better to have an approximate answer to the correct question than an exact answer to the wrong question. And these p-value methods are approximate answers to the right questions. Just a couple of quick things that we're done. We're getting less and less technical. That Cauchy was kind of the test your merit. So how do I combine, I've got 17 different studies, all sorts of different ways, maybe different tests, et cetera. How do I combine that information together? This is the field of meta-analysis. Appendix 6 talks a lot about that, but just in a couple of slides, here's the basic idea. So here are your studies. In your studies, here's the actual value of the parameter you're trying to estimate. There's your estimate. Then you have a sample size, a standard error p-value, and you may have all these other moderator variables, for example, sex, age, et cetera. So with SNPs, you may have data like this. You've got estimates, the SNP effects over different studies. You've got sample sizes, standard errors. Again, why don't we combine p-values? Because we're more interested in not whether something is overall significant. We really care more about what effect it has. So meta-analysis is largely framed not in terms of p-values, but in terms of estimating effect sizes. And Appendix 6 talks about some of the justification for that. There's a pretty robust literature on that. So you have fixed, random, and mixed model meta-analysis. So fixed effect is really simple. Theta is your true parameter. You assume it's the same in all your studies. Ti is your estimate in study I. It's got an error. That error, you know it's standard error because it's been reported. So your estimate of theta over the samples is just simply this weighted average here. This is weighted least squares. There's your overall weight, and there's your standard error on that estimate. So really straightforward to do. That's a fixed effect. You assume all the thetas are the same. Well, in biology, we often expect the thetas to vary. SNP effect could vary depending upon, maybe, for example, it's tagged well in study 1, but tagged poorly in study 2. So that actual effect, the marker effect, is going to vary. So the Cockrum Q-test for heterogeneity is simply given by this form here. It's a chi-square, and this simply asks, is there excess heterogeneity across your samples? When that occurs, you can use what's called back up in a second. So we can take this fixed effect, we can also add on these other things. I'm trying to estimate the SNP effect, but it may be affected by age and smoking, et cetera. You can add all those things in here. This now becomes a standard fixed effects multiple regression. The other framework is random effects. So again, there's my error, but now my theta is some overall value and then some specific realization for this specific study. And we assume that that realization has a mean of 0 and a common variance. You can also think about this as theta I, the sum of these, has a mean of mu, which you're trying to estimate, and this variance. The estimate is exactly as you've seen before, t divided by the weights, but now the weights become this. The weights now depend upon these variance among the mu's between studies. You can estimate that with this estimator down here, which is a Rimble estimate. So the main difference between a fixed and random effects is, and again, you can add more things. This will give you a mixed model. There is your fixed effect, there's your random effect, write it that way. The main difference between fixed and random effects is both of them have this estimator. In fixed effects, it's here. What does it mean? It means more precise studies are given more weight. Under random effects, the weights are now given by this. So if this between sample variance in your treatment is large relative to this, under random effects, all studies get pretty much the same weight. So random effects may be a more biologically realistic model to look at how those theta estimates vary over samples, but it comes at the cost of all the studies become weighted more equally because it's not the precision that matters, it's the precision plus the fact there's variance over studies because this term varies. So that's fixed and random. I think I'll stop there. Just a couple more slides you can look at. penalized regressions, AIC, BIC, pretty straightforward stuff, but I think you guys have held on long enough. And again, the notes are posted. Okay. So questions or comments? Taking you up to dinner, aren't I nice? Well, I can take you up to dinner if you want, but I think dinner's at 7, so. So key takeaway. Don't worry about the details, you know, what's a Cauchy, don't worry about that. The key takeaway is you now have realized that, number one, if all the p-values are from a bunch of tests are all from the null, it's a uniform. That allows you to get an estimate of the fraction of all the things you test at that actually are from that. We talked about how do you control for multiple tests. How do you do that multiple comparison? We talked about Bonferroni ways. We talked about false discovery ways. We talked about Bayesian ways. We've talked about resampling permutation approaches. Then we talked about meta-analysis combining results from different studies. So in the field of GWAS, almost all human GWAS now are done by meta-analysis. And they've been really clever. And one of the reasons they've been clever is because of privacy concerns and also, you know, some proprietary information. I can't send you all my data, but I can send you my estimates. If I can send you my estimates, I can combine them. And they've shown basically that combining the estimates is essentially as good as putting all the data together and analyzing it as one. And by combining the estimates, you can account for differences in methodologies over different studies. And again, there's lots more stuff that Appendix 6 talks about that on meta-analysis. Okay. Great. Thank you for hanging in there and good questions. And we will see everyone tomorrow.